Welcome back to the HIV RNA Test Guide podcast. We're your trusted source for staying updated on all things HIV, especially reliable testing across the U.S.
Great to be here.
Today, we're diving into something pretty exciting. Potential news in HIV research?
Absolutely. It involves an investigational therapeutic vaccine candidate, HB 500.
That's the one. Developed by HOOKIPA Pharma and Gilead Sciences. Now, this is important for everyone listening. People living with HIV, families, healthcare folks, researchers, here in the U.S. and really globally.
It's definitely got people watching.
Okay, so here's the thing. The actual Phase 1b trial, it's still ongoing. We're expecting the main results later in 2025.
That's right. So no official data just yet.
Exactly. So today we're exploring a what if.
Sure.
What if the early signs from this trial are, really positive, exceptionally promising?
The hypothetical deep dive basically helped us understand the potential.
Precisely. What is HB 500? How's it meant to work? And what could hypothetically good news mean, especially for folks in the U.S.? Plus, what's the road ahead actually look like?
Because it's never just one step, is it?
Never. Our mission here is just to unpack this potential development. But, strong disclaimer, this is all based on a hypothetical positive outcome. For the real facts when they come, you absolutely need to go to the official sources.
Always check the official clinical trial data.
Okay, so let's get into it. To really get why something like HB 500 could be big news, maybe we should quickly recap where things stand with HIV in 2025.
Yeah, setting the stage is important. What's the challenge we're still facing?
So, HIV human immunodeficiency virus. It's a virus that goes after the immune system.
Specifically, those CD4 T cells, the helper cells that coordinate everything.
Right. And without treatment, HIV just damages those cells over time,
Yeah.
weakening the immune system.
Until you reach the stage known as AIDS, acquired immunodeficiency syndrome, where the body can't fight off infections.
But treatment has come such a long way. Antiretroviral therapy, RT, that changed everything.
Absolutely revolutionized it. Daily meds suppress the virus, stop it from copying itself.
And people on effective ART, taking it consistently, they can live long, healthy lives now. It's become a manageable chronic condition for many.
A true medical success story. Phenomenal progress.
And that leads to UU, right? Undetectable equals untransmittable.
Crucial concept. If your viral load is undetectable on RT, you can't pass HIV on sexually.
Which has been huge for public health, but also for reducing that awful stigma.
Immense impact on both fronts. It really shows the power of effective treatment.
And this is the big but, RT isn't a cure.
No, definitely not. And that comes with its own challenges.
It's a lifelong commitment, taking pills every single day. That pill fatigue is real.
It really is. And adherence is absolutely critical. Missed doses and the virus can come back, maybe even become resistant.
Plus, even the newer drugs can have side effects, short-term or long-term.
And then there are issues like cost, access, especially for some communities here in the U.S. Consistent care and monitoring aren't always easy to maintain.
And sadly, even with UU, the stigma hasn't vanished entirely.
No, it persists, and that affects everything from mental health to seeking care. These are the reasons why we still desperately need a cure.
So why isn't RT a cure? You mentioned the virus coming back if you stop.
Exactly. It's because HIV sets up shop, basically. It creates viral reservoirs.
Hiding out.
Sort of. It goes dormant inside immune cells in places where ART just can't get to it, so if you stop the meds...
The virus wakes up and starts multiplying again from those hidden spots.
That's the core problem. The latent reservoir is the main barrier RT can't overcome.
Which is exactly why researchers are pushing for a cure, or at least a functional cure. And that brings us right back to therapeutic vaccines.
Yes, like the HB 500 candidate we're discussing.
Now, it's important to make a distinction here. We're not talking about a preventative vaccine, like for measles, which stops you from getting infected in the first place.
Right. We still don't have one of those for HIV, unfortunately. Decades of research. But HIV is just incredibly tricky. Mutates so fast, hides from the immune system.
So a therapeutic vaccine, like HB 500 potentially is, that's for people already living with HIV.
Correct. It's designed to modify the existing infection to boost the body's own immune response against the virus.
And the big goal. Often it's aiming for a functional cure, or sometimes called sustained ART-free remission.
Meaning, ideally, someone could stop their daily RT.
And their own immune system, trained by the vaccine, would keep the virus suppressed at undetectable or very low levels for a long time, maybe indefinitely.
It wouldn't necessarily mean every trace of virus is gone. That's a sterilizing cure, which is even harder. But it would mean managing HIV without daily meds.
Imagine that. No more daily pills, fewer RT side effects, maybe lower costs, just better quality of life.
It's a massive potential shift, letting the body control the virus itself.
So how do these vaccines try to do that? What are the strategies?
Well, there are a few main approaches. One big one is boosting those killer T cells, the CD8 plus T cells.
Like HB 500 aims to do.
Exactly. Training them to find and destroy HIV-infected cells, including maybe cells in those reservoirs.
Okay, what else?
Other strategies focus on antibodies, maybe stimulating broadly neutralizing antibodies, or BNAbs that can hit many different HIV strains.
And I've heard about kick and kill.
Yes, that's another angle. Trying to wake up the dormant virus from the reservoirs, the kick, so that a boosted immune response or another drug can then eliminate it, the kill.
Lots of different angles being explored then. Different technologies too.
Oh yeah, DNA vaccines, RNA vaccines like some of the COVID ones, viral vectors using harmless viruses, protein-based, cell-based therapies. It's a really active field.
So developing a successful therapeutic vaccine, that's a huge goal. And any sign of progress, like potentially good phase 1 data?
Generates a lot of excitement. Understandably so.
Okay, let's zoom in on HB 500 itself. We said HOOKIPA, Pharma, and Gilead Sciences.
Right. HOOKIPA specializes in arenavirus-based immunotherapies, and Gilead, well, they have a long, strong track record in HIV.
A good partnership, potentially. So what's unique about HB 500's tech? You mentioned arenaviruses.
Yes, it uses two different ones, Paschen virus, or PICV, and lymphocytic choriomeningitis virus, LCMV.
Okay.
They've modified these viruses so they can replicate enough to deliver their payload, but they don't cause disease in humans. They're replication competent but non-pathogenic.
And the payload is HIV components.
Exactly. They're engineered to produce specific HIV proteins antigens. So when these vectors get into cells, they make HIV antigens, and that trains the immune system.
And the training is focused on?
The platform is specifically designed to generate really strong and hopefully long-lasting CD8 plus T-cell responses, those killer T-cells we talked about.
Gotcha. And which HIV parts are they targeting?
They're using antigens from conserved regions of HIV-1 clade B.
Clade B, that's common here in the US and Europe, right?
It is. And conserved regions means parts of the virus that don't change or mutate as much. The idea is to get an immune response that works against many variants.
Makes sense. Using two different viruses, focusing on conserved regions, aiming for strong T cells. Sounds like a well-thought-out design.
It addresses some key challenges. The use of two different vectors, PICV and LCMV, in sequence is also thought to potentially enhance the immune response further.
Okay, and the Phase 1b trial, NCT 064-30905, that finished enrollment back in January 2025.
Correct. It's looking at safety, tolerability, any immediate reactions, and immunogenicity. Does it actually trigger an immune response in people already on suppressive RT?
With results expected later this year, 2025. So now, back to our hypothetical. Let's imagine the early buzz is really good.
Right.
What does exceptionally promising actually look like from a Phase 1b trial?
Well, first off, safety is paramount in Phase 1. So good news absolutely means an excellent safety profile. No serious side effects linked to the vaccine. Maybe just mild temporary things, sore arm, bit of fatigue, ache stuff you see with other vaccines. It has to be well tolerated. That's baseline.
Okay, safety first. Then what?
Then the big one, immunogenicity. Is it doing what it's designed to do immunologically?
Generating that T-cell response.
Exactly. You'd want to see clear evidence of strong CD8 plus T-cell activation. Researchers would measure this, a significant increase in T-cells that recognize the HIV antigens from the vaccine.
How do they measure that?
Using lab tests like ELISpot or flow cytometry, they can count the reactive cells and see what they're doing.
And it's not just about numbers, right?
No, quality matters hugely. Are these T-cells functional? Can they produce antiviral signals called cytokines? And crucially, can they actually kill HIV-infected cells in lab tests, in vitro?
Okay, so quantity and quality of the T-cell response.
Yeah.
Anything else?
You'd also look for early signs that the response is broad, might work against different HIV variants, and durable, meaning it looks like it might last.
Now, what about actually impacting the virus itself? Is that something you'd see in Phase 1b?
Usually that's more for Phase 2, especially since participants are already suppressed on RDA. But the absolute dream scenario in Phase 1, maybe some subtle early hints of an effect on the virus, perhaps a slightly bigger drop in the latent reservoir size compared to placebo. Highly speculative at this stage, though.
Or maybe in a special sub-study, like an analytical treatment interruption and ATI. Though you said that's more phase 2.
Correct. ATIs are complex. If they did a small, controlled ATI and saw that people who got HB500 had a slower viral rebound off OT, wow, that would be huge news, even if very preliminary.
Okay, so any sign it's affecting the reservoir or controlling rebound would be massive.
Absolutely massive, because hitting the reservoir and enabling ART-free control, that's the ultimate goal. And HB500's approach, using these potent arenavirus vectors for strong CD8 plus T-cell responses.
That's exciting because those T-cells are thought to be key for tackling the reservoirs.
Precisely. If HB500 showed it could safely do that in early trials, it would really validate this whole strategy.
So let's run with that hypothetical. Imagine HB500 does show these strong positive signals. What are the ripples? The impact, especially here in the US?
Oh, the impact would be profound on so many levels.
For people living with HIV here.
It's just immense hope. The real possibility of an ART-free future. Freedom from daily pills, from that fatigue.
Reduced treatment burden, fewer side effects, maybe fewer clinic visits.
All of that. Improved quality of life, physically and mentally, less anxiety about adherence, and, you'd hope, another blow against stigma.
Okay, what about the healthcare system?
Long-term, you could see significant cost savings, less spending on expensive ART, potentially fewer hospitalizations, managing fewer side effects.
And for the science world.
Huge validation for HOOKIPA's arenavirus platform, maybe opening it up for other diseases like infections or even cancer. It would likely accelerate research and funding across the entire HIV cure field.
Like a rising tide lifting all boats.
Sort of, yeah. And crucially, we'd learn so much more about what kind of immune response actually controls HIV, which helps design future therapies.
And globally.
Yeah.
Beyond the US.
Massive implications. HIV is a global pandemic. A successful therapeutic vaccine here could eventually offer hope worldwide, especially in places where lifelong RT access is tougher. It could be a vital tool for ending the epidemic globally.
So this hypothetical breakthrough, it's not just science, it's a massive beacon of hope.
Absolutely. Transforming lives, reshaping the future of the pandemic.
Okay, but let's bring it back to Earth a bit. Even with amazing hypothetical phase 1b results, it's not like it's available next week, right?
Not even close. That's where the realism has to kick in. The path from phase 1 to an approved treatment is long. It's a marathon.
So after phase 1 comes...
phase 2 clinical trials, bigger group of people, maybe several hundred.
What's the goal there?
Still checking safety, but also figuring out the best dose and schedule and getting a much better idea of efficacy. Does it actually work to control the virus? This phase can take months, often years.
And this is where those ATIs, the treatment interruptions, would likely happen.
Yes, very likely. Carefully stopping RT under close watch to see if the vaccine controls the viral load compared to a placebo group. Plus, continuing to monitor immune responses and safety very closely.
Okay. And if phase 2 looks good?
Then comes the big one. Phase 3 clinical trials.
These are the huge ones.
Yes, large-scale pivotal trials. Thousands of participants, often across many sites, maybe internationally. Really diverse population.
And the aim here?
To definitively confirm effectiveness and safety compared to standard care or placebo. This is usually the longest, most expensive phase, several years typically.
And this is where many drugs stumble.
Unfortunately, yes. Phase 3 is the make or break stage. Lots of promising candidates fail here, either not effective enough or unexpected safety issues pop up in the larger group. The failure rate historically is high.
Wow. Okay, so if, hypothetically, HB500 makes it through phase 3 successfully,
then the companies, HOOKIPA and Gilead, can submit all that data preclinical phase 1, 2, 3 to regulatory agencies like the FDA here in the US.
And what's called a BLA, Biologics License Application.
Correct. And then the FDA's experts, doctors, scientists, statisticians conduct an incredibly rigorous review. They scrutinize everything. Safety, efficacy, manufacturing quality.
How long does that take?
Many months, sometimes longer. They often bring in an independent advisory committee of outside experts, too.
And even after approval, they keep monitoring.
Absolutely. Post-market surveillance or phase 4 studies continue to track safety and effectiveness in the real world.
So, bottom line, even with the best possible phase 1b news, we're talking years.
Realistically, yes. Maybe 5 to 10 years, potentially more, before something like HB500 could be widely available if it clears every single one of those hurdles. It's a tough, rigorous process for a reason.
Patient safety, public health.
Exactly. It also highlights that HB 500, even if successful, is just one piece of a much bigger puzzle.
Right. The whole landscape of HIV cure research. It's not just this one approach.
Definitely not. We have to remember the huge challenges HIV throws up.
We'll remind us again.
The virus's incredible diversity, constantly mutating, the latency, those viral reservoirs hiding out, HIV's knack for evading the immune system, and, historically, not having clear correlates of protection, knowing exactly what immune response guarantees protection or control.
So HB 500 is one promising shot.
Within a huge global effort, there's so much else going on.
Like gene therapy. I remember hearing about things like EBT-101 using CRISPR.
Yes, investigational gene therapy is trying to actually cut HIV out of cells. That's one avenue.
And those broadly neutralizing antibodies, BNAbs, using them as therapy.
That's another major area, infusing powerful lab-made antibodies. Plus, other therapeutic vaccine candidates using different platforms, and those kick-and-kill strategies we mentioned.
So it's likely the answer might involve combining things.
Very possibly. A combination of strategies might be needed for a functional cure. It really takes continued innovation, solid research funding, global collaboration.
And people willing to participate in the trials.
Absolutely crucial. Trial participants are true partners in this research.
Okay, so bringing this all together, our deep dive focused on a hypothetical positive scenario for HB 500. It really highlights the hope in HIV research right now.
It does. There's a lot of smart science being applied.
But the actual HB 500 trial is ongoing. We wait for the real results later in 2025 from HOOKIPA and Gilead.
The whole community is watching closely.
And regardless of this one trial, that search for a functional cure presses on. Driven by researchers, funded by various groups, and powered by trial volunteers.
The dedication is remarkable.
And for everyone listening, your health, your awareness, it's paramount. If you have questions, if you're unsure of your status, if you need testing,
knowing your status is the first step.
Please visit our website, hivrnatestguide.com. That's hivrnatestguide.com. We've got reliable info, resources, and access to confidential, lab-certified HIV RNA testing at over 4,500 centers across the U.S. Getting tested is taking control.
It really is. Information and access are key.
So what do you think about therapeutic vaccines? What are your hopes, your concerns for HIV cure research? Let us know in the comments. We really want to hear from you.
Keep the conversation going.
And if this deep dive helped you understand things better, maybe gave you some perspective, please give it a like. Share it with others who might benefit, and definitely subscribe to the HIV RNA Test Guide podcast.
Hit that notification bell, too.
We'll bring you more discussions, news, and insights on HIV research, testing, and everything related. Thanks so much for joining us on this exploration. I'm your host from the HIV RNA Test Guide podcast.
Until next time.
Stay informed, stay healthy, and we'll catch you in our next deep dive.